All right, so you're back. You know how we do things here. No time wasted. Straight to the good stuff. Today's deep dive is all about how certain antibiotics knock out bacteria, specifically by messing with their ability to make proteins.
That's right, we're focusing on protein synthesis, that fundamental process all cells need.
Exactly, and we've got a ton of scientific info on how different classes of these antibiotics work. So basically, we're going to break down their mechanisms of action.
Yep, we're going to pinpoint how they manage to attack bacteria while mostly, leaving our own cells unharmed.
Because that's the key, right? How do they differentiate?
The big difference, the vulnerability we exploit, is the ribosome. Bacteria have these 70S ribosomes, while our cells have 80S ribosomes. And that's where these antibiotics come in.
Okay, so let's jump right in. First up, aminoglycosides. You've got your gentamicin, streptomycin, the classics.
Right, and they all go after the smaller subunit of the bacterial ribosome, the 30S, specifically a part called the 16S rRNA.
16S rRNA, and what do they do there?
Well, they're pretty aggressive. It's not just one thing, it's like a triple threat. First, they cause the ribosome to misread the mRNA, like the genetic blueprint.
Oh, so like a typo in the instructions.
Exactly. It leads to the wrong amino acids being added to the protein chain.
Meaning messed up proteins from the get-go.
Uh-huh. And then they can also make the ribosome just stop prematurely. Like, okay, we're done here, even though the protein isn't finished.
So you end up with these incomplete, basically useless protein fragments.
Right. And on top of that, they can actually block the ribosome from moving along the mRNA. This process is called translocation. Imagine the ribosome as a little machine moving down a track. Aminoglycosides basically throw a wrench in the gears.
So it's a complete shutdown of protein synthesis.
Yeah.
And that's why they're bactericidal. They actually kill the bacteria.
Exactly. Multi-pronged attack.
All right, moving on. Next, we've got the tetracyclines like doxycycline, minocycline.
These guys also target the 30S subunit, but their approach is different. Well, they bind near this site called the A site, which is where the tRNA carrying the amino acid is supposed to dock.
The tRNA being like the delivery truck, bringing the building blocks, right?
Yeah, perfect analogy.
Yeah.
So tetracyclines basically block the loading dock. No more deliveries.
So the protein chain can't grow any further. And that makes them bacteriostatic, meaning they stop the bacteria from growing but don't necessarily kill them.
Yep. Like hitting the pause button on construction.
Makes sense. Now, let's talk about macrolides. Erythromycin, azithromycin, these are pretty common.
Yes, and they go after the larger ribosomal subunit, the 50S. Specifically, they target a region within the 23S rRNA, close to this important enzyme called peptidyl transferase.
Peptidyl transferase, that's the one that actually creates the bonds between the amino acids, right?
Got it. And they also mess with the exit tunnel.
The exit tunnel, that's where the newly made protein leaves the ribosome, right?
Exactly. So macrolides can either block the tunnel altogether or just make it harder for the ribosome to move, to translocate.
So the protein gets stuck inside, or the whole process just slows down significantly.
Right. And this usually leads to a bacteriostatic effect, similar to tetracyclines.
Interesting. Now, our sources also talk about lincosamides, like clindamycin.
Yeah, and these work in a pretty similar way to macrolides. They also target the 50S subunit around the
Right.
same area and also interfere with peptide bond formation and translocation.
Oh, so they have almost the same mechanism.
Yeah, which is why you sometimes see cross-resistance. If a bacterium develops resistance to one, it might also be resistant to the other. And like macrolides, they're mostly bacteriostatic.
Okay, got it. And then there's chloramphenicol, right? Another 50S inhibitor.
Yes, chloramphenicol binds to the 50S subunit, but it directly blocks the peptidyl transferase enzyme.
So it just shuts down the bond-making process altogether?
Pretty much. It's primarily bacteriostatic, but it can have some serious side effects, so its use is limited.
Makes sense. Now let's talk about the oxazolidinones, like linezolid. Our sources say they have a unique target.
They do. Oxazolidinones bind to the 50S subunit, but at a different spot, near where it interacts with the 30S subunit.
So at the interface of the two subunits.
Exactly. And what's interesting is they actually prevent the very first step of protein synthesis, the formation of the initiation complex.
Ah, so before anything even gets going, they stop it right there.
Yep. And because they have this unique mechanism, they tend to have less cross-resistance with other antibiotics. They're mainly bacteriostatic.
Okay, and finally the streptogramins, like quinupristin and dalfopristin. These work as a pair, right?
Yes, they act synergistically on the 50S subunit. Dalfopristin basically helps quinupristin bind better.
So they boost each other's effectiveness.
Exactly. On their own, they're usually bacteriostatic, but together they can be bactericidal. They inhibit peptide elongation, and can even cause the release of incomplete peptides.
So it's like a 1-2 punch.
Exactly.
Okay, so what we've seen here is that there are all these different antibiotics, and they have various ways of attacking the ribosome.
Right, but the overall strategy is to target the differences between bacterial and human ribosomes.
And exploit those differences to shut down protein synthesis.
Exactly. It's all about stopping that protein factory.
Which raises an interesting question, I think. If these drugs can target bacterial ribosomes so specifically.
It really makes you think about how essential ribosomes are to all living things, and what challenges we might face as bacteria evolve new ways to resist these antibiotics.
That's something to ponder, for sure. All right, folks, that's our deep dive for today. Thanks for joining us.
Thanks for having me.